well, and of course so once again, welcome to the Institute, welcome to the workshop. I hope you have a good time and go back a little bit more, having learned a little bit more than what we know in the morning. So in the evening, you can ask the question, what is it, what delta do you know more than you did in the morning? If you have a positive delta, and hopefully it will be positive, it can't be negative. Well, actually, if there's some deconditioning, then it may even be negative, which also is in the end sometimes positive. But as long as you go back with the fruitful delta, which is college, I think you will feel good. That's something else. We will feel good that we will. So thank you for attending. Thank you for my colleagues for arranging the workshop. Thanks.
Thank you, Director Gupta. Thank you, Radha, for organizing this meeting. I was very fortunate that I started my scientific career in the laboratory that was established by Professor G. N. Ramachandran in 1974 at Molecular Biophysics. And all biochemists, microbiologists, and all this thought, oh, these guys do nothing, they punch cards and rotate molecules. And that was the beginning of informatics in biology. And Professor Ramachandran understood it very well. Computational biology cannot tell you what is correct, but can definitely tell you what you should not be doing. What will not happen, it will be eliminated. And Ramachandran map is nothing but an exclusion map. Infinite possibility of transformation that protein can take. He said, this march is not allowed. And it turns out, today, every single protein crystal structure that is being solved, they fall within the allowed region that Ramachandran done. So I think this is the biggest masterpiece of informatics application to biology ever in the history of science. And it happened to be a person from southern part of India. When Human Genome Project began, I knew that it would be difficult for Indians to participate in sequencing. But I was very sure, and I have a cartoon, I didn't know I'll have this time to talk, but I will show the cartoon, which I wrote, which I drew in 1990s, saying that experimentalists will find this will be like a blind man looking at the elephant. You will get this, you will get this, you will get this, but somebody has to integrate this in computer. This huge amount of data has to be informatically understood, comprehend, and there needs a Francis Crick who will say, hey, there is a need for a tRNA which never existed, and he said there has to be one adapter molecule for protein synthesis. And I always do this picture with an Indian photograph, Indian sitting there. We used to go jog Ramu, remember? We used to say Ramachandran or Raghavan, somebody we do. All my South Indian boys do IT better sometimes, than they do. So I used to say, one of those guys will do. Question was it be done in Indian institution or will it be done sitting in California or Germany? So therefore, we focused while building IGIT to have a strong informatics component. The second thing Indian genome could contribute is patient sampling. Because China, because of one-child family, do not have large number of children per family. So India's failure in family planning is an asset for genetics, those who don't understand genetics. Because you have to collect information. The larger the family, the better is the information, you can collect with respect to eliminating opportunity and getting it more focused. So therefore, the genome sequence, today we have, I think, 650,000 genome getting sequenced, million will be soon, and XSC database is huge. But what is important is not individual information, but what is important is how do we look at multi-layer information, layer after layer, and put them together, and come up with what we call system biology. So future lies in system biology. The next information technology revolution has created IoT, Internet of Things. Like I know exactly how much I walked yesterday and how much I walked today. You have your sleep pattern, all these digital devices are collected. My formal talk I'll end with telling where is the future going. The future will be not to treat a patient, but to remain healthy. And to do that, we need to collect digital data of our lifestyle, along with genetic data, biochemical data, and what we have learned now, that all of us carry about 2 kilograms of bacteria in our stomach. And this bacteria actually decides what you have eaten and what is the metabolite you produce, whether you are likely to be diabetic or asthmatic, or you are going to have a psychological problem. This is new. Just imagine if we are talking about 20 or 1,000 human genes, we are talking about 1 million bacterial genes that are playing inside your. How the hell you are going to do this experiment unless you explore this proposition? So I'm very glad to see that you are getting equipped, this institute. I had a dream that we should have an institute in Delhi, other than something down south in Bangalore. How to bring integrated people. I can see people like here, Raghav has joined, and I congratulate the director for acquiring such extraordinary assets, including documentation of curriculum. So you have fantastic sets of faculty, and therefore one needs to explore the multi-dimensionality of the biology. You see, understanding ourselves, when the anatomy was understood, people thought human body is understood. Then came the biochemistry, then came the genome, then came the proteomics, then came metabology. Now we have a huge overlay. People haven't tried. Why do somebody gets a cardiac problem with a lifestyle? Like Indians have 2.2 millimeter arteries, whereas some Caucasians have 3.2 millimeter arteries. So if there is an Indian who has 3.2 millimeter artery and you have taken his genetic map, still he will not be risk because his artery is bigger. So how do you integrate in biology a risk program from molecules to another? And that's going to be next 20 years of science. So I think this institute is poised very correctly, has positioned itself. And we need to, and this is, I can see the open source leader is here. I believe in open source. And I'm sure the open source leader will take it further. So with this I can say, this, I think awareness among students to understand things in a totality and not just IT to IT or biology to biology, but to integrate. So therefore I say there is a great opportunity in workshop continues, and I'm very happy to contribute whatever you want to contribute. But I can only say, I wrote my first paper using only informatics in 1979, okay, when we had only 29 protein structures solved, and Professor Shashekharan came back from Princeton with output of 5-side map. Just output, because he brought the output, and looking through the output, and then deciding which residues will favor beta tau and which residues will not. Those days our punching card was there, right? There was no. So I think I look like grand old person right, Vishwa Pitamaha, who believed that you can get a scientific output out of a technology which was considered to be a supporting technology. I'm sure Raghava started his career like that. So, but today he is a leader in the field. So I would say that begin anywhere, but must remember, Nobel Prize will come in physiology of medicine to one person who will never do a single experiment, but will come up with fantastic ideas and solutions based on biological informatics. With this, I hope one day one of you will aspire for such laurel to drink, because the opportunity is finite. I wish I was 25 today. Then I would have been a run for it. Thank you.
Thank you, Professor Kamasari. I would now request Dr. Sriram to talk about the MTech CV program.
So, let me begin by saying a very warm good morning to all of you. In the next 5 or 10 minutes, briefly I am going to talk to you about the activities of Center for Computational at IIT. First, it was conceptualized in the year 2015. Initially, the institutional support towards the center was around 11 lakhs per year. And most importantly, we signed the MOU with CSIR IGIT in order to leverage their expertise. Because the Center for Computational Biology initially started basically as only for the computational purposes, particularly working on algorithms, then working on basically mathematical modeling, then developing software. In order to leverage the experimental side of it, we signed the MOU with IGIT, and it was signed by Professor Rajesh Kothari and Professor Jaloti in April 2015. And in June 2015, within a month or two, it was inaugurated by Professor Vijayaraghavan, Secretary DBT. Presently, Professor Raghav is the head of the Center for Computational Biology. And most importantly, under the auspices of Center for Computational Biology, these are the educational programs that we offer basically. The first thing, most importantly, IIT is a research-led institution, so we give more importance to research, as well as the other educational programs. So we started with a PhD in computational biology. Current strength is around 5, and followed by the MTech in computational biology. The current strength is basically 10 in the first batch and 9 in the second batch. And we have also started, we started earlier prior to MTech and PhD, we started BTech in minor in computational biology, particularly for the CSE and the ECE students. And as Professor Jaloti mentioned, that we have submitted a proposal in the year 2015 itself for the financial support for MTech program. And now it was reviewed favorably, and also it has been funded for us for 3 years. And it's going to start, our funding starts in the year 2007. That is in another couple of years. Okay. So what is the mission of this particular Center for Computational Biology? Of course, to conduct basically the outstanding educational research, basically because most of the faculty comes basically from the academic background. So initially we want to start with an academic research, and later on we want to foray into basically the industrial consultation. So mostly through this activity we want to promote, stimulate and educate basically the computational biology programs and the academic research, as well as to do industrial consultation. So the mission is to be the leading center in the country for developing advanced mathematical and computational techniques for the biological system, particularly for the informatics part of it. With the advent of new technologies, we generate a lot of data And to analyze this data, we require basically not only the computational skills but also to develop new software and also build models to understand the biological functions with existing data. And also, we want to have a partnership with the industry, research and academic laboratories so that we do have a good network of not only from academic side but also from the clinicians, medical researchers and the academics. Okay. So what are the activities that we have taken for the last 2 years? Regularly we are conducting workshops and seminars and also an outreach program that we have, like a workshop like this or the seminars like this. And we also conduct weekly computational biology seminar series, and it serves various purposes. Particularly, the speakers are invited both from research organization as well as from the industry. We got the speakers from this organization particularly to have a research exposure, because we also would like to know that what other people are doing in the country, particularly to know about the recent advancements in the computational biology. And the collaboration possibilities both for the faculty members and also for the PhD students. And we also actually have students going and doing the thesis and internship with the other institutes. And also to attend the students from other places for doing the MTech and PhD programs. And we also invite speakers from industries, particularly for the placement opportunities, because we are running the MTech program. So we also want students to be placed in good industries. So we call those people to interact with the students and to do internship and thesis jointly guided by the faculty here and also from the industry. And to have an industrial exposure and to have a potential computational biology profiles and also the work options for the students in the industry. Finally, most importantly, that like the entrepreneurship and the startup ideas based upon whatever the academic research that which you are doing, whether it's possible for us to convert it to a startup program. And besides that, it's also the consultancy opportunities. And who are the core faculty? Because all these things cannot be possible without having a good faculty. And Professor Rama, you all know, so there is nothing for me to explain because he is one of the leading bioinformaticians in India. He is presently the head of our cell for computational biology. And Rebecca and Bebo, they are basically working in bioinformatics and next-gen sequencing. Rebecca is particularly working on algorithms for bioinformatics data, and also Bebo is working on basically the graph algorithms, Epigenetics data, and next-gen sequencing. Ganesh Praglers, Shubhadeep and Sriram and myself, we are working on basically the systems and network biology, the area of systems and network biology. Kabujjoti Pera and Rajiv, they both are actually computer scientists. They are basically working on algorithms and machine learning, and Kaupitya Shetty is also working in machine learning applied to healthcare problems. So, the most important program which works under the CCB is basically the MTech in Computational Biology program. It consists of three very important parts, namely the computation, mathematics and biology. The four pillars of biology, namely genetics, molecular biology, cell biology and biochemistry, we cover it thoroughly under this biology. In mathematics, we cover a lot of things like stochastic process, linear algebra, statistical computation. And in computation, we cover specifically algorithms and programming. So all these things are actually packaged into, all these three things are packaged into the computational biology. This is the first of its kind master's program in India. This is specifically only for the computational aspects of it. As I told you that we signed MOU with IGIT, so we leverage most of the experimental data, try to leverage the experimental data from the IGIT. And now we are not restricting only the IGIT, but also all the other CSIR institutes and DBT institutes across Delhi all over. So, what is our program structure of MTech computational biology? Most of the people who are coming from coming and joining this particular program, they requested that they should have a preparatory courses prior to the starting of the program. So to accommodate their requests, so we started the refresher courses for like molecular biology, data structures and algorithms, crop spans and linear algebra. These are some of the courses which we started as a preparatory module, and this preparatory module runs for a year, for a month. Sorry. And we also provide a coursework of 32 credits, and 16 credits goes for the core courses and 16 credits for electives. And besides these 32 credits, we also give 16 credits for the thesis. So the computational biology program in MTech is overall 48 credits. And also, as I told you earlier, that like we do also have a weekly seminar series, which is mandatory for most of the computational biology. Besides MTech program, we also run basically the PhD program in CCB. As I told you, there are 5 students presently working in various areas. So we don't have any restrictions for the PhD students' background. They can be from any background, whether they are BTech, BE, MTech, MS, any background. We are willing to absorb them and we are willing to train them. So we also take students from MSc in other science disciplines who are eligible for PhD. So if you want to know more about the eligibility criteria, please look into this website. Thank you. If you have any questions, you can shoot me or you can send an email. You can look into this website and we are willing to, we can answer your questions. Thank you.
Alright, so now inauguration part is over. So we will start the session. And the first session, we have 2 speakers, Dr. Brahmachari and Manish Kumar. That's very interesting. One side is my mentor, other side is my issue. Okay, so I will not talk in detail about the Dr. Brahmachari achievement because it is too much. He got most of the awards, whether it's Bhatnagar Award, JC Bose Award, so I cannot count all the awards. He is fellow of all the academies, FNA, FSC, FSC, FNSC, so even Third World Academy, so he has a lot of it. So I cannot go in the detail. And he was PGCSIR, he made the IGIV. So in all terms, there are a lot of things. So I will not go for that, and I will not keep this time. Okay. Only thing I want to say 3 things actually. I know he was building the organization. He built the IGIV, built the CSR. This is a big achievement according to me. Another part is basically love towards science. I meet a number of people. I have not seen any person who has so much love towards the science. I remember a long time back we organized one workshop, international conference in our institute. He was busy CSR at that time, so busy. He was secretary, a very busy person. But he was there for 4 days, he listened all the lectures. That's a love for the science. I have not seen any person. And another thing, mentoring the youngsters. He always looking for youngsters to talk to them, to convince them. He is mentor. I got his mentorship from last 3 years actually. So he forced me to expand my scope from simply developing simple software to involved in OSDD and other major projects. These are the 3 things according to me. And anyway, I will not eat this time. So Professor Antibiotic.
We'd rather ask come and give this talk, I was wondering in this hot summer, I'm very reluctant to travel. And I was until yesterday in Kashmir, enjoying beautiful weather, and he said 16 to give this no hot. But as I thought, what I will do this exercise of telling you how one actually goes about, need to go about in informatics today. I have played my 3 innings of my scientific career for the younger generation. I started my career as a biophysicist when I did all the structural biology work, where I did experimental work extensively. Then 1997 I moved to Delhi from Professor of Indian Institute of Science to Delhi to build Institute of Genomics, where we did Indian genome variation and also built a genome informatics where I met Rapa. And then I was moved upstairs, forced to be away from science, doing active science, but I managed to do the open source drug discovery project and continued this activity. Since I retired, I realized that I can do science. I don't have to continue to do a science management, because I learned one thing, that it's very difficult to advise older people. And as a DG, you only advise the directors and senior scientists. You don't really get access to the younger. But I managed through ISDD. So I decided that I don't want to do this. I quit at 62, so that before I become too old to go back to science. Today I'll talk to you how we have used system biology and informatics in actually moving forward in a different way. And of course I spend a lot of time on wellness genomics and affordable IoT, Internet of Things. I mentor a few companies. I help them in big data analytics for health. I also have a startup on genomics last 1.5 years. The genomic data deluge is huge. In 1990, when the Human Genome Project was promised, we had an idea. From this sequencing of human genome, we were able to solve all problems of all diseases, including asthma, diabetes, cancer, cardiovascular, of course, infectious disease. Unfortunately, the explosive growth in biomedical data generated has not yet translated to proportionate clinical data. The reason is, much of the data, when the data is small, experimentalists can handle it. When the data runs into few thousands and thousands of pages of Excel sheet, experimentalists feel hopeless. So data organization, data structuring, data capturing, then data analytics becomes more important. System biology is nothing but capturing all structured, unstructured data and converting them into meaningful understanding is system biology. Therefore, the challenge lies today is to develop what we call system medicine. If biology is like a plant, medicine is like a forest, system medicine is an ecosystem. So the forest doesn't exist, and I was sitting in an ecosystem where the snows are melting, roaring river, little river coming down in Pahalgaon, the beautiful valley, that's an ecosystem. And this ecosystem we don't understand. How do we compute lifestyle? How do we compute all biochemical changes taking place with the genetic makeup and our daily food habits? All this integrated together is actually the system medicine. And when we can understand that, we can actually design precision medicine. 20 years back, 1998, Mycobacterium tuberculosis genome was sequencing was available. But the problem was, even after 10 years, there was no drug. I asked myself, if I have to build a human in computer and treat the human, that is, put everything we know about a human genome, human pathways, human biochemistry, everything you put together, and then say, okay, let me now add salt and increase the blood pressure, simulate. I realized that there is not enough data. And it may take 30 years of data collection before it can be done. And I was already getting old, so I didn't believe that I will live till 30 more years. So therefore, although genome is projecting that we may live up to 110 years, can I take a smaller number of genes? So I asked myself, can I pick up Mycobacterium, 4,000 genes, instead of 20,000 human, and human, the network of interaction goes into factorial, not simple numbers. So 4,000 and 20,000 is astronomically different. Okay, those who do network analysis, they'll understand. So the idea was this, and this led me, actually under this 2002, we had a project on in silico biology, and one of the objectives was to build a system biology model platform by integrating data into it. It originated when we published the Indian genome variation work. Ramachandran said, sir, can we not do something, Dr. Ramu, with IGIB, when we will have 100 author paper? I said, 100 author paper, just like Indian genome variation. Let's do some computation. So that was the origin of SysBot, System Biology of Organism project. Unfortunately, within IGIB, doing that, just to say why TB? Because TB kills a large number of people. Even today, 1,000 people will die every day in India. So I came up with the idea that within IGIB, we tried actually here with lots of project assistants, collecting data, 25,000 pages of data we captured. But we realized it's still the most drop in the ocean. So I came up with the idea, can we do an open source approach? And I show this slide to say, this is 20th September 2007. It is just 2 months before, 1.5 months before I became DG. So I got the idea, I went around, I raised some money from the government, got an approval from Mashelka, but unfortunately I became DG after 6 weeks. So it became the CSIR program rather than IGIB led. Although the project was approved. What is interesting, when I presented this, and it was, I think I presented in the UK, and it was picked up by some Mint journal, that is the Wall Street Journal, and this is William Hasseltine, which is the richest biophysicist in the world, who was a Harvard professor and founder of the Human Genome Science Project. He instantly wrote to me that they what a great idea, and asked me to join for a conference. And in India, you can't get a letter on 20th September, email and say 5th October come to Washington. It is not possible. Right? I said, thank you very much. So what I say that when he said what a great idea, I knew I don't have to worry about anybody else. So therefore I didn't bother. Let's understand what is this intellectual property and associated, why drug discovery is difficult and expensive. So if you want a healthcare benefit access to all, it should be available. If it is available, then only IP becomes. So if you want the product to be available, intellectual property increases availability. But as strong intellectual property, decreases affordability. So Raghav's open servers increases availability, right? Whereas Schrödinger's software decreases accessibility. So if you want affordability and availability to make accessible to all, we need to have a balanced view. So we need to create open innovation and shared wisdom. Personal genome sequencing was available at $300 million in 2003, but it was not affordable. So the price of sequencing has fallen to $1,000, but the drug discovery has gone to $2 billion now. Why? Why is it so? So this is the question. There's something wrong. what is wrong in our drug discovery when sequencing has become cheaper and cheaper. So, it is the innovative technology. If you look at, people will tell you that clinical trial all the cost. Let me show you the clinical trial. 87% of the late stage failures are due to poor efficacy and safety, which means a drug should have never entered the clinical trial as entered the clinical trial. Phase 2, phase 3. So it is waste of money. 87% money to be saved. 70% fails at the late stage. You need to do only 2 clinical trials phase 3, but look at companies doing hundreds of clinical trials at phase 3 for a drug. Why? The reason is the drug is not working. And you don't want to drop that drug and say it's not working. Then the company share goes down. So 20 years back, a transformation happened in pharma company where scientists were leaders, suddenly finance people became leaders. And once finance became leader, they're not interested whether science is moving better or not. And that's how, if you know the drug called Gleevec, it was done clinical trial with only 58 people. When an 8 feet tall person comes, you don't have to measure whether he's tall or not. So when a drug is outstanding, it just ends out. So therefore, but I have convinced myself, I can save 87% of the failures by combination. So that was the argument. So what is the problem with tuberculosis drug? Because the convention is you invest, you patent, new products are produced, investment comes, return of investment to the market, and increased involvement of R&D, the circle goes on. But lack of market incentive for neglected disease break the traditional innovation cycle. So neglected disease will never get an opportunity of this cycle. And the proof is, during this period, 100 plus cancer drugs have come. Whereas 1963 is the last drug, repurposing, is in the market. In 2007, we had nothing. And it's only 2007, when a lot of noise was made on TV, Bill Gates Foundation and the support, and eventually build up like we did is in the market for drug-resistant TB, and new drugs are being tried. So this is where we began. The concept was to collaboratively aggregate all the information that is available in literature, bring them together, and build a model, and then do computational analysis. And this, you know, more than 8,000 students and people participated in 480 countries. So system biology, in the case of an organism, is the application of drug to uncover all pathways, all reactions of a body. Can any student tell me how many reactions you think takes place in a Mycobacterium tuberculosis to make one bacteria to do it? Anyone guess? A guesstimate? Students, no facts. Nobody can guess. How many? To make the cell wall, to make the DNA, to make the protein, to make this. We have 4,000 genes. Let's assume 1/4 of them are responsible, let's guess. And out of this 1/4, then we'll do how many reactions? Young man. Tell me, you're sitting with that now. Yes. So actually it was not possible to guess, because there was no literature to guess. So, the objective is, what is system? System engineering is well worked out. IT has done a fantastic job. Today, Airbus 380, one of the prototypes is moving to Paris Museum. Why? Because it was never made prototypes. It was all made in computer. It was flown in computer, unlike Wright brothers when you made trial and error. And then the prototype was manufactured. Today, Formula One cars are not manufactured and tried. Formula ones are simulated. 100 simulations, best 100 simulations are then taken, and 5 of them are models are made, which are tested in wind tunnel. And after testing in wind tunnel, only 2 are actually manufactured, and 1 of them is driven and wins the race. So all this is engineering, this is computation. If this has happened then why are we in the Wright brothers era for drug discovery? Try and fail, 5,000 molecules tried. So system structure, system control, system design, and you need system dynamics to understand. So this is what I said, the drug discovery fails because it's a very complex network of interaction. So when you try to make a molecule which will interact here, it can also interact with something non-specific. And this problem goes on. So efficacy to increase and toxicity to decrease is the objective. So how do we then compute this in? So the chemists have gone on making molecules and miserably failed. And they feel very frustrated. 5,000 molecules you make, then one molecule at a time. It's too expensive. And actually we have done the experiment for 5,000 molecules. It's too expensive. So can we reduce computationally to 1 in 100, 1 in 20? That was my dream. So we needed 45,000 research papers to be read. And as I told you, 40, 30, 40 summer trainees were doing few months work. I realized I will be 100 years old by the time this gets done at IGIB. It will never happen. So therefore, we made sure we open it up. And what happened then, we got lots of students, and this is known to all of you, that we used this community, collaborative community, and got a 99.5% annotation of all 4,000 genes. 3,952 genes were annotated, and this Vinod and Anshu played a big role. We used virtual cloud workspace by connecting Google Doc, and Raghava also responsible to see that the mirror site doesn't collapse. And he contributed to large amount of tools parallelly were getting built. And this data was captured using Twitter, and all people were correcting each other. And we used every possible social media to do this exercise. The students didn't have access to laptop, so many of them actually did this. And one student would say, after reading a paper, that this gene is for this function. Other student writes back saying, no, it is not correct. Then there is a dispute builds on, and on that dispute, then Anshu or Vinod and somebody sit down and say, hey, it's okay, we found the solution. So in this way, this was completed. So a lot of structure, unstructured data, and I tell you what students' power is. 3 students decided that they will be responsible to get all the papers which are not easily open source paper. So that means you email to the author. That means hundreds of emails are going every day. Those papers are retrieved, put into the archive, and the students can access that. And that is how more than 50% papers have been brought by the email access. There were the students set up what I call a call center, round the clock. So that any student needs help for annotation, they are available. And all this was done. I had participated in few hundred Skype, along with Anshu and Vinod, and eventually we could build the entire data. Students were brought in Bhuti Bhavan, the top 120 students who did excellent, and we had, I think that's the only time you have seen BGCSI wearing a t-shirt of OSDD, and I preserved it very well. And then the world noticed it. The world noticed it before even we published it. And that's a fantastic part of science, this science, that it had more publicity than ever I have written something. You write then publicity. I did not have a thing to cite our paper because we haven't written the paper, right? We have written the process. And everybody is writing papers in Nature and everywhere and talking about it. And of course, Zalil and team did a wonderful job of cutting down my presentations into various YouTubes. So power of social media. Without publication, OSDD became very famous. Now what did we do? We created an open architecture, and Infosys came and helped us, and Raghava was one of the captain along with Debashish, Vinod and others to make sure that they are doing the right job so that we got a professional and everything was open source. And this allowed us first time to publish some papers, and we decided that we'll get all them published in open source journal, structure analysis Mycobacterium. We built the networks, and based on network analysis, we identified 73 central important proteins. These proteins are not reacting, they're interacting. And from this, we subtracted from the sequence level human genome human gut bacteria and oral flora, and then structural level peptide conformation analysis, protein binding site level, we need to look at similarity. And after all this filter, if you ask me, we were left with only one which fulfilled all filters. So it was very, then we said, okay, little bit filter cut karo. So, so from 3 central protein, we landed up about 17 molecules, proteins, that looks to be very, very important for the biological network. And these 17 we published. I'm rushing through because it's published. So next one I want to cover what is just now we have finished. And you can see what was interesting, PK and B, and some of the genes which are already in the target PKD are actually fed into our. We built the biochemical pathway network and figured it out that, but most of them are transcription factors. And biologists have a lot of difficulty in assessing transcription factors. Transcription factors are not reactions, so that you can build an assay. Transcription factor assays are difficult. Some people are trying, we get an idea, but it's still not completed. I suggested that you look at transcription factors binding site. Based on this, can you find a gift in structure and see whether overlapping transcription factor site can you block with a small molecule so that transcription factors will not bind. This interaction can be blocked. So the interaction between a transcription factor and a protein is pretty tough. Okay? We can analyze it, but to design a drug to assay is very difficult. So this is where the challenge lies, and I can tell you this is still an incomplete challenge, and this remains. But the next challenge, so we could publish this, and it was high hit. And what was interesting, we built a network of young people, around 45 individuals are co-authors in this paper, and half of them I never met physically. And I have organized a workshop in which I met many of them, right? He organized in Chandigarh, we met many of them. And this is interesting from Germany as well as from Malaysia, students participated across the world. Those who contributed more than 1% became author, and those who contributed less than 1% became a member of the consortium. So I bring to you, this is what happened. Still I was in the CSIR, of course we finished the paper, and then I detached. Then I saw the data is seeding, but nobody is asking that question, how many reactions takes place in Mycobacterium? Because you need to understand biology, a lot of biology. So I had a student whom I picked up from OSDD, so this is my crowdsourced student. He was one of the smart kids who was actually doing OSDD leading projects in structuring how you should annotate. But he was a computational kid, and he was actually a mathematics lover, and he didn't understand biology very well. So we had a crash course of him that he had to read everything in biology. And 2013 was dedicated to him to read biology if you want to do PhD with me in 2014. So he had to take only biology courses. He was not allowed to take computational courses. And Rohit in 2013 January and Rohit in 2014 summer was a different person. Today he is in Stanford. Okay? So what did we do? We looked at the system. Already the data was available, and we could see 890 genes involves 961 metabolites to do 1152 reactions. Mycobacterium metabolism. Okay? If you look at what was existing information available, the dark blue is already annotation was available. All these additions of genes we did in our new annotation. These pathways were completely missing. We got this new pathway. It was only 36 pathways earlier. We became 50 pathways. And you can see the number of metabolites, the percentage of reactions, everything started growing in our, that is we call OSDD, OSDD. Okay? He loves all these names to give. So 890 genes is now our story. Okay, you put them out. This is the picture you get. I'm sure no computational guy will say, I want to work on this. Am I right, Rahul? And that's why everybody was sitting. So I didn't know what to do. This is not computationally amateur, right? But each is linked to each other. Everything is linked to each other. Why I'm telling you this story, that how you think is very important. So I realized that none of the resources I have around are actually able to build a computational model of this picture. That's exactly the metabolism of Mycobacterium. It's terrible. All poster, we can bring a poster and take it to the lab. Every biochemistry lab will have such poster, right? But we don't make a sense out of it. We have been seeing this from 70s, right? You spend your whole scientific career in corner of this map to study one area, or you corner of this map of one area, and these are one lifetime of a faculty with 20 students. Correct? So how do you make it easy? So I was lucky. I got 4 IT students from IIT Madras, IIT, NIIT, 2 NIIT and 1 triple IT, 2 from Bhubaneswar, 1 from Madras and 1 from Epigenetics, 1 triple IT. And they knew nothing about biology. So I told them, you don't have to worry about this. So I gave them first month trying out, but they didn't succeed. Then I told them, you know the Dabbawalas in Mumbai. What do they do? Anybody knows the story of Dabbawalas in Mumbai? Raise your hand. They deliver dabbas, right? From home to the office, through all possible paths, and with a Six Sigma, no failure. It is so confidential that you can put your checkbook inside it and reach the office without any problem. So what do? They label the dabba, right, and transfer. Each dabba has a transfer location, destination location, pickup location, and they go transfer depending on the lines. So 400,000 dabbas moved with Six Sigma accuracy. So there are lines. So what is chemistry? A metabolite is a substrate, reacts with an enzyme to produce a product. That product is a substrate of a next enzyme to make another product. And that product is a substrate. And that's how the membrane is built, that's how the DNA is built, that's how the whole metabolism goes. So I told them, can you write algorithm for this? They said, this is no issue. This will write. And they wrote the algorithm in the next 2 weeks. Okay? Movement of dabba. Now I say, now change the dabba into metabolites. Okay? Now let's see where, so I think it was my, then I decided that I will convert this into a 4-axis problem to, there is something happening outside the cell, something happening inside the cell. Inside the cell, some metabolites are interacting with genes, and then genes are creating reactions and pathways. So can we put them into 4-axis issue? And this is how it was put. So I told, now you take this, as if they are drill lines, and the stations you are changing the metabolite. So connect, there are some only small numbers, 76 or so outside, like sodium, potassium, water, they move across the membrane. So that's not a big issue. Big issue is this. So you take 961 metabolite, 890 genes, and 50 pathways involving 1152 reactions. So take each metabolite, connect to the gene, take it to the pathway, and then thereby the metabolites which are being used by many genes to many pathways, put them in the middle, and then less and less ones to the outside. Genes, you order them according to replication origin, gene 1, gene 2, gene 3, so they're across, there's no issue. Because in biology, prokaryotic biology, reactions are very well organized across the genes. And then you put them into pathways, the most number of reactions are here, the most used metabolites are here, and things are like this. So this was not difficult for them. When this was done, the picture emerged like this. Now you will say equally complex, right? But remember, this is now in computer. So I know every metabolite along the axis. I know every genes along the axis. I also know every reactions along the axis. Of course, your first question was, what the hell these reactions are? Can anybody tell? What are these reactions? Bacteria has spent so many enzymes and so much energy into it. and you realize it is a defense mechanism. Where does India spend its maximum budget? Education? Where does India spend its entire budget? Maximum budget.
Defense.
Science?
Defense.
0.89%. Health? Less than 3%. Defense? 18 to 22%. Mycobacterium spent the same. This is defense. Sidney Brenner, I learned 1981 one thing, that in biology, when you don't understand something, look at society. If society has behaved this way, then biology inside the cell behaves the same way. Right? This is I learned when I was very young. So this must be defense, and it just turns out oxidative becomes. I'll show you what type of reactions are out. So let's say, can we change the media in computer? And says yes. I don't put 961 metabolites, I just start reducing them. Middlebrook media uses only small number of metabolites, and that happens to be about 284 metabolites, is sufficient chemicals to have the reactions to go, and you can see now bug is happily growing, and what you measure is a flux balance, you measure the biomass at the end, but what happened to the defense mechanism? No. You know, you sit in Delhi, you don't see military, you go to Kashmir, you only see every corner one military standing, right? So, we don't use militaries are in the barrack. Mycobacterium militaries are in the barrack, where it is happily growing. Clear? So what is this situation? So we said, now I can do computationally. I know only 400 genes are essential and involving 439 reactions. Now let me knock out one gene, which is target lysine. Once I knock out this, what happens? Some reactions go, some of the metabolites are no longer getting processed, and the moment I do this, you can see the consequence. The reactions have dropped, 292 reactions have gone, mass is not produced, bug is not growing. So now we have a computational tool by which I can knock out gene by gene by gene by gene. Right? I can knock out metabolites, I can do whatever I want to see how is the reaction taking place. Then, but I tell you, this needed huge amount of my mind time, which I could not have done if I was teaching CSI. I can think and figure it out and to make students do it. But what was surprised, when we knock out that gene, suddenly 60 more genes get activated and 50 odd reactions starts becoming, some new reaction starts happening, 57 new reactions. So bug starts responding. The moment you put drug, and it turns out you have blocked one pathway, it opens up another pathway. Right? And you can see that. So we plotted them, and we want to see experimentally this is happening or not. So therefore literature data clearly shows that 60 genes we are talking about, at least 48 of them we could detect. Experiment. So therefore our approach computationally is working. Then we looked at the drugs which have already become resistant, and you can see that the more rare it is, the worse it is. The more less it is, the better it is. By this approach, we could say the CAT-G gene is working, and then we set up this box of genes, actually it is shutting down and not creating too much of, whereas these genes, which drugs are already there, are all becoming highly resistant. This is where the interesting part, when we discovered that ApoE, the target for beta-quinine, and then this is PPE1, which is the new preclinical trial going on, suggesting that our approach is working. We looked at the lower concentration of the protein, and based on knowing that the lower the concentration of the protein, the lower drug we will require, one could inhibit, and then from this, out of 116 essential genes, we could actually show 70 of these are actually essential, some are persistent, and then we suggested 4 pathways. These 4 pathways, when we put all this in the metabolic adaptation pathway, we found that those rare genes that come up to prevent, they actually, what is happening, these are the rare genes which I listed, is actually against new genes are expressed. So what's happening when you are blocking this mycolic acid synthesis, then the reduction of triglyceride in this cycle, the de novo synthesis NADH takes place here, And this NADH1 is inhibited by metformin. So therefore we suggested type 2 diabetes drug metformin could be used along with the drugs to prevent persistence and latency and resistance eventually. I take 5 minutes, it's okay. Okay, these many drugs are fine. So we have a huge database of Mycobacterium genome database, and that database has been curated about 1623 genome, of which that data had 1084. When we plot these 890 genes, the white is all mutations, synonymous mutations. You can see they're all clinical strains, and these are the 890 genes. You can see huge amount of mutations are taking place in children. So our principle was, okay, the target is fine, these are good, but what if it is mutated in the strain? Then it's not going to be functional. So then you can see that my essential genes of 116, how many of them are already hugely mutated are no good. Similarly, persistent genes are also mutated. I went back and checked my 17 genes into this database, and you can see the PKNP, why it is not working, because most of the biobank pathway is mutated. So hope is PHPMP, C-gauge, and these 3 are potential, 3, 4 are potential, rest are finished, you can't do anything. So therefore, evolution is an important component. Based on this, we found out 25 genes which are not at all mutated in multi-drug resistant bug, and these 25 genes are. Then we asked, how come the experimentalists have made a little mistake? And they have made sequencing error. Why the hell are we believing that they are correct? So we allowed one sequencing error per genome, because we have sequenced, and then we got 8 more genes into the kit. Now what happens here? What was interesting, the targets of beta-quinine and all this came up, including my metformin, which are untouched. And these are the 8 more genes we got, and we believe these are the ones. We looked at their druggability assessment, and they're highly druggable. We looked at the crystal structure of them, and based on this we found structural analysis. So what we could do then, up to this, we have published. This is gone for publication, working on it. Repurposing FDA drugs, novel 3-line metabolite, understanding multi-drug resistance, identifying this. So one part we have completed is by doing virtual screening of molecules, and from the virtual screening molecule and 776 GSK molecules which have been given active, we did virtual screening, and this is well within all of you bioinformatics part. Then shortlisted the RVD, 7 crystal structures, and looked at their GSK molecule. And the one thing we discovered, the most of the bioinformatician handle, when they get the genome size, one sequence, then they suddenly go to PDB structure. PDB structure sequence, crystallographers have changed it. Because in order to crystallize, they changed it. So in the process, you're actually handling a wrong molecule. And this is what the mistakes are. So we made sure, Mukta is here, she made sure that all are, so I had a postdoc, Heath and Mukta, and then all these corrections, so you get the biochemistry target, you identify the crystal structure, we get the molecule, this all of you know how to do, I don't have to tell. Then from this we identify new molecules, and we suggest the molecules. Then we link through the molecular dynamics, and then eventually we are proposing 6 drugs which are in the market, metformin, lansoprazole, and 4 new molecules, including tetrapositine, and this is lemonoprid and roxaprigal. Repurposed drugs which will work on various targets, and dipopredine, another one, and some additional molecules. Now these are all docking studies. We also looked at the highest mutated gene, and it turns out second highest one is happened to be a protein which is ISJ2, CAT-G for isoenzyme resistance. We wanted to understand why this is causing disease. Then we got all the drugs, existing drugs mutation, and you can see very badly pyrazinamide, thambutol are hugely mutated and unlikely to work for a long time. I want to close here, but since you said future, I'll take 2 minutes to say future. So one of the idea of preventive, predictive, personalized medicine, I showed the crowdsourcing, drugs repurposing is an opportunity for you. Lots of opportunity in cancer and many of them using the same system approach. Then there is a crowdsourcing patient wisdom, which is done through traditional knowledge data, which is sitting there. We haven't integrated. And then wellness initiative, big data analytics, sequencing 100 years old people, which we have done about 100 of them at IJV. With FitMate and IBM Watson Health, artificial intelligence is the future. So future, Raghava will walk like this. And all this data will get sold by him. And he will do artificial intelligence to say, what is the likelihood of Bhargava getting a cardiac arrest? Right? Out of the students' disastrous research that some student has done, right? So that he thinks so. So this is how will be the world of tomorrow. And this data we need to collect. And I have initiated this activity to what is called IoT of health. Now if there's a P4 medicine continuum, I'm saying we are a reactive world. After you disease we treat. Can we be a proactive world? Where before you fall sick, we treat. But the weather of Delhi, you can't do too much of exercise, right? I was average walking 10 to 12 kilometers last 5 days, 6 days. And suddenly I know today AC is moving. So this, according to me, I have not published this yet. This is my proprietary, I have put the activity there. Rather, you can see the modern medicine is trying to see health, to biological symptoms, to clinical expression. Actually, go to doctor at stage C. When you feel giddy, when you feel weak, when you get chest pain, that's the time you go to doctor. We are saying, modern medicine is saying, go to doctor at this stage. Right? So keep profiling. Then you bring back here. Don't have to go to D1 disease. Ayurveda is telling, actually you are a normal individual, the changes starts taking place. And then it expresses into the various tissue. That means your cholesterol has gone up, but not deposited. After deposit only the blood flow will decrease and pain will develop. So this is Prokapa and Prasada is the where you stop. If you've already gone to this stage, Satta Samadhara is already disease has symptoms. Modern medicine actually calls one stage of disease. Ayurveda defines 2 stages. One stage where the disease has happened, but is reversible. The disease happened which is irreversible, like peripheral neuropathy, retinopathy and all. And this is where, so I started with one of my students with philosophy of learn from the data, involve patient in care, each patient data counts and keep healthy people healthy. And we have been able to create data collections, and this is called because we wanted to look at child mortality. In India, it's pathetic that 40 children die in 10,000 but US is only 6, even Philippines has reduced it to 20. And our target is, India's target is 30. So this is how we have done. Since you are talking about making money, so this student of mine, we worked. Last year we launched the products. This, we received at least 90 lakh rupees award. Award, and then we have $120,000 cloud time from IBM and $120,000 cloud time from Intel as award, and we have been able to spin off the company, and the company has got its first investment of 9% with a disposal of 1.5 crores for 9%. So it is possible. But you have to be entry early. We have entered the field. We have so far 40 small children born. This is the Dhirubhai Ambani Hospital, Kokila. So we have picked up the expensive ones so that we can get them. And then we have also got the poor hospital in Rawari, and this data is under investigative analysis. So this data is getting, just to tell you, an ICU child generates 1.7 gigabyte data per day. And the child stays for 8 to 12 weeks in the hospital. Just imagine the data. Can we predict? So we have created an app. App is given to the patient from that, those parents. And can capture and say, why 25% children die ahead of time? Can we predict that this child is entering into sepsis phase? Can we prevent? So there is a great opportunity. So I shared the work of Heath and Heath. So I'm no longer DG, I cannot say my team. So my team is very small, and the decibels and Gupta is taking forward. Heath is now a staff in UK, and Rohit has gone to Stanford. So thank you very much.
Excellent work.
You told that metamorphism can be a potential weapon. So can we achieve that on diagnostics?
Okay, so you are right question. So the one of the first thing we should do a crowdsource. Before I could do the crowdsource, a diabetic patient with TB, Singapore has a cohort, and they showed that they have already got a TB patient diabetic cohort who has taken metformin, and they are getting, but it's a very small step. They have now the other group has now done experimentally induced mice with diabetes and treated with normal drugs and metformin. TB clears. So ICMR DG is taking it up. As you know, OSDD, I had a slide to show that what ICMR DG is doing. I better publicize that, otherwise she will get upset. How I missed it? Just sorry, I just saw this. So, as I said, the journey, now the government of India has taken up TB Research Consortium under Stop TB. It's called Indian TB Research R&D Consortium. ICMR is taking it. All OSDD data has been taken up by them, and metformin clinical trials are starting. Even nation has already declared TB by global agenda. Open Source Pharma Foundation Trust. So what we started is getting completed, and OSDD has gone global. I don't know how I skipped it. Even Bill Gates in 2007 did not, when I asked Mashelkar, can you go to Bill Gates and get some fund for OSDD? Bill Gates said, never go. And Mashelkar was having a TV, but television interview with Bill Gates, and still he said, no, shall be done. Bill Gates now says open source collaboration between scientists would be the best way. So Mashelkar retweets and says this. So you know, and I just want to say, you know, in life, you do some science, you do it, as I say, you get an award. When I was young, I was bothered about awards. When you grow old, you don't bother. A book has come, published by Mark Stevenson. He visited us in 2015 when I was in the IGIB as a JC Bose Fellow. And this book tells about 13 global innovations which will change the world. And one of them, he covers the story of OSDD. So it's very gratifying to see that something like this has happened. And I thought young students will feel motivated. But sitting in India, when you say something new, they will ridicule you. People will make fun of you. And he had absolutely captured those in the story. It's a beautiful story. I will give you a book I just got. I didn't read it. It's available on Amazon. So this is what I say, a cast of characters who are by turns inspiring, demanding, and driven. So it's an interesting book. You have many stories, including Patient Like Me, if you go to the website. The guy who started Patient Like Me with now 400,000 patients of Alzheimer, Parkinson, and all are on the website. And they themselves are doing clinical trials. They don't care about the pharma. They are bypassing the pharma. So all possible innovations are written there. So I thought it's worth reading. And I was very happy to say, OSDD became one of those. Thank you very much.
Any other questions? Otherwise, time.
Thank you. I'm here. Lunch time.
Thank you very much. On behalf of Okay, Mani, definitely. I always enjoy them. So I will like to see the second thing is how campus we did PhD after me so that we did master from GLU, understand? GLU master. Postdoc from Canada. He is the only person of my friend who was not interested to go abroad. He want to work in India. So he was probably 1.5 years. 2 years. Canada. Got the opportunity and joined that. Yeah. So you are talking about that? Maybe. Yeah. 10 o'clock.
Thank you, sir, for providing me opportunity, and it's really humbling for me that I have to give my talk after Professor Brahmachari, and I have missed all the excitement around OSDT because when it was started, I left MTech. Although initially I worked for approximately 2-3 weeks, but then somehow I lose interest and I did not continue. So today I am going to talk about my work, one part of my work which is dealing with antibiotic resistance using in silico tools. So now how antibiotics is important for India. In India, the burden of infectious disease is the highest among the world, and you will be surprised to know that almost 50 lakh children die from the two infections, that is Streptococcus pneumoniae and Haemophilus influenzae, which can be easily prevented by the vaccine. And due to coming up of antibiotic resistance, this burden is going to increase. And due to antibiotic resistance, the people have to go the treatment for a longer time and they are at more risk of dying. Also, in case of epidemics, it is very difficult to contain that. So the mortality rate will increase, and also the person who are giving care to other persons, sick person, they are at greater risk of infection. Now there are 4 main mechanisms of antibiotic resistance. The first is altered target site. Here this is antibiotics, so it goes inside the cell, pathogen cell, and pathogen somehow alter the target, and the antibiotic can no longer bind to the target. The second is decreased uptake. So pathogen modify the channel through which antibiotic penetrate inside the cell, and the drug no longer can go inside the cell. Then there is increase in efflux, so the antibiotic can able to enter into the cell, but it has been spit out. Then there is a bypass pathway, that pathogen somehow make a bypass network, and it can bypass the original target which is on which the antibiotic was targeting. And then there is enzymatic modification of the antibiotics, where antibiotics can go inside the cell, but it can be enzymatically modified. So till now I am working at this aspect, that is modification of antibiotics by some enzymatic mechanism. And there are many causes of antibiotic resistance, like particularly in context of India, there is lack of microbiological facilities. So whenever we go to any doctor, they don't have any way to check what antibiotic he should use. So they give the broader spectrum antibiotics, and in that way the resistance develops. Then doctor prescribe the patient, any patient who has a fever, they simply give the antibiotics. It may not be a bacterial infection. Then patient expectations are there. Like when you go to a doctor and he does not give you the medicine, it means doctor does not know anything. And then desire of pharmacist and some doctor to make profit from the drug sale, and then lack of knowledge about the inappropriate use of antibiotics. They routinely pop up antibiotics without bothering that what it is doing to our health. So the data, as Professor Brahmachari told, that there, in case of Indian countries, the data is very limited. We don't have data. We tried very hard to take some data. So the maximum, in case of this, so this is a CDC study which was published in 2007, and it says that even in US there are a lot of misprescription of antibiotics. So it is not only in India. How this antibiotic susceptible pathogen turn into a antibiotic resistance one. So the first picture come in our mind with this question that it must be the hospital, but the fact is not that. It is the agriculture, the animal husbandry and the fishery which is the main culprit behind that. So these three fields consume the majority of amount of antibiotics, and through which it goes into our food chain, and then ultimately it comes to our body. So this gradual accumulation of antibiotics ultimately feed up all the susceptible microbes. So our gut has mostly the antibiotic resistance. Our gut contains lot of antibiotic resistance genes. So how to limit the drug resistance? So there are very casual, there are very simple way by which we can reduce the burden of antibiotic resistance. The most foremost is antibiotics should be used only when it is necessary, and it should be taken with proper care. That it should a patient should complete the whole prescription. And whenever possible, take the vaccine instead of using the antibiotics. So now coming to what I work. So as I told you that I work on beta-lactamases. It is the most widely used group of antibiotics, and it is a broad class of antibiotic consists of beta-lactam nucleus in the molecular structure, and it inhibits the cell wall. It inhibits the cell wall formation, and there are many generations of beta-lactam antibiotics, that is cephalosporin, Mycobacterium and carbapenems. And beta-lactam work by inhibiting cell wall synthesis by the bacterial organ, and it basically cleave this bond and make it ineffective. This bond is cleaved by the beta-lactam antibiotics, and that's why it becomes ineffective. So beta-lactamase is basically an enzyme which does this function, and it is classified into 4 different classes on the basis of Ambler. There are 2 different ways of classification of beta-lactamase. Ambler system is there, and then Bush-Lie system is there. So this is basically a class A, B, C, D is on the basis of sequence homology, and this Bush classification also includes the hydrolysis profile. Now coming to what I have done in this. So I have a collaboration of Professor J.S. Heath in Department of Microbiology, and he has a very huge collection of Yersinia, which is a enteric bacteria, and he was basically he routinely does MIC test to see what is the extent of antibiotic resistance. So when he tried to analyze this table, he told me that look, we have this result and we could not find that what is the reason behind that. They were suspecting that okay, this can be a upstream region modification and modification in the coding region. So he get it sequenced, and as you can see, this is the upstream region. There are few regions, this is absolutely conserved. So there is no region behind that modification in upstream region contribute to differential hydrolytic profile. Then if you see the protein, there are some modifications. So we did some docking experiment, docking and modeling experiment, and there also we could not find anything which is contributing to this differential hydrolytic profile. Then we were basically stuck. Then somehow we tried that okay, let's look into the mRNA secondary structure profile. And what we found that in each group in each different biomarker there are differential structure of mRNA. So at that time we proposed that okay, it may be the region and it can be a potential region behind the differential hydrolytic profile, but we could not validate it. But later some report came which basically validated our hypothesis. So thus, there is another work which I took with in the collaboration of Professor Dr. Deepa Bist in the Jalma Institute for Microbial Disease. So here we did as you know that over 100 million children are vaccinated each year with BCG, and the laboratory attenuated variant of which is basically attenuated version of Mycobacterium bovis. Although the efficacy of BCG vaccine is highly debatable issue, but it is everybody agrees that it does provide some immunity against the TB. And there are many studies which has studied the differential expression pattern of BCG in 5-osome and all these things, but there was no study which has done the comparative study of this BCG inside the macrophages. So for that we have taken two different strain of BCG, MTB bovis, BCG Danish strain, and then there was a cell line BPH1, in which then we did the 2D gel electrophoresis and MALDI, and we find that there are few proteins which are differentially expressed in that, and these are the proteins. So we are now we are trying to biochemically characterize these proteins and look at whether we can target these or not. So this is all the work which I have done with in collaboration. Besides that, in my own lab we also do mostly bioinformatics stuff. So, and in each beta-lactamase ampler class there are few residues which are highly conserved, and they are mostly the active site residues. So we try to find out that is it the only conservation which beta-lactamase has, or is there any another conservation which is attributing its particular function. Like all the family in a class does not have the same hydrolytic profile, they have a different hydrolytic profile. So there must be something which are attributing that differential hydrolytic profile. When we analyze that, then we saw that this hydrolytic, beside the active site, there are other regions which is highly conserved. So using that, we are trying to expand this work in a different direction. Then we have also developed a database of beta-lactamases. So this is a very comprehensive database. You can do searching using keywords, and then with keywords you will get a lot of information about the beta-lactamases, like what is the hydrolytic profile, this signature, where it is bound, how many variants are there, what are the mutations are there. So few simple things are there. There are a lot of papers and other curations are also there. So there are many other beta-lactamase databases, but beside we have incorporated all the features, but in our databases there are the features which is not available in other databases. We have also developed a classifier for classification of beta-lactamases and its handler class, and recently we also upgraded it to the next level. Like in class B beta-lactamases, there are 3 subclasses, B1, B2, B3, so you can classify further. One of the recent projects which we took is the development of point-of-care diagnostic device for the collection of antibiotics. So the objective behind this project was to develop a point-of-care device that can test a clinician, that can tell a clinician that what should be the proper antibiotic he should employ for the treatment of patient. So it will have long-term benefit, like the test will enable the physician to detect AMR and prescribe the most effective antibiotics to a patient within 30 minutes when patient lands. This 30 minutes is the most crucial window in any clinical setup, because this is when a patient needs the antibiotics most. So at this stage, most of mistreatment occurs, because when a patient comes with some accident or something and the doctor has no option available with him, they just give any wide spectrum antibiotics. And also if this type of POC device is available, it will also stop all empirical prescription of antibiotics and allow quick control of the infections. So our device tried to develop a test which integrate the technology and using a microfluidics platform, and it is basically a microarray chip with a microfluidics platform, and it will tell what is the first antibiotics a doctor should give, what is the second line, what is the third line. So this is what we have proposed. So this is our platform, and we are hoping that test would directly recommend the most appropriate antibiotics for the treatment. And ideally it should be within the 30 to 45 minutes of sample collection. And it would also eliminate the hit and trial, which is the most of the time is responsible for the death of patient. That a doctor is giving the antibiotics against which resistance is all there in the antibiotics in the patient. So in Indian setup, another very crucial factor is the affordability. The test should not be too costly, too expensive. So this is our schema. So you have a patient sample, do the cellular lysis, digest and pass it to the platform, and it will give you that okay, this is the suggested first line treatment, second line treatment, third line treatment. So we propose this platform to the NESTA, which is a non-governmental organization of UK, and it funds this type of point-of-care devices along with the Innovate UK. So we have got the grant, and our proposal was selected. There were total more than 100 proposals, and 12 were funded. And there are only 2 government organizations which were funded. Rest all are companies. So this is not what we have done. There are other prediction servers. We are also doing other aspects, like we are also developing prediction servers. We are also developing databases. Recently, my main focus on the lab is this metagenomic and with commercial aspects. So in metagenomic, we are trying to correlate between the low complexity region and disorderliness of protein. Basically, we are trying to harness this in terms of catalysis efficiency. And then we are also doing analysis of gut microbiome, metagenomics, and construction of their interactive networks, so that we can find out that what are the potential bacteria which we can target. and how this antibiotic resistance basically disseminate even the gut microbiome. We are also doing comparative analysis of viral metagenomes and their role in antibiotic spread. This I am doing with Dr. Rajiv Paul in Department of Microbiology. In another commercial aspect, this project is almost done and we are in process of drafting the patent. So we have engineered the PCR sumo protein to enhance their protease activity and optimization of production capability. So this is our team, and the team which is involved in antibiotic resistance. Professor J. Smith is there, Dr. Manisha Goyal, Nidya Singh, and my students in my lab, Dr. Ravinder Kumar, Vandana Kumar, Yadisika Srivastava, and Diksha Pandey. And I should also acknowledge Professor Painter because he is the one who actually drives us. So whenever we are down, chalo yaar, chalo main tumko coffee pilata hu. Then he tells about a story of his GM Brassica कि देखो ये भी होता है, ये भी होता है, ये भी होता है, ये भी होता है. So you should not get disheartened at this age. I think I have finished earlier. Quite fine.
First, we should thank you for saving time. He didn't know that we are nice. So, as a grandson, I will give you in science, if you are asked 10 years from now, tell in 3 sentences. Assume the textbook is written, or the citation is written. In 3 sentences, so my suggestion, you learn all the skills, all your boss's skills you have, I can see. Sit down and decide. 10 years later, what 3 sentences you can write in your life? That is all I've done in my life.
Sir, this.
I've seen all of them.
No, this thing. This is a suggestion. No.
Sit down and ask, will it be relevant 10 years later? Will, I can tell somebody, I always say, when I tell my grandchild, they ask, what have you done? Like academically. So I can say 3 sentences. So you don't think 10 years, because you have all the skills. When you develop skills, because you actually have integrated all this very well. You have in front of you a fantastic opportunity. Ask a question, answer it. This will make it. Figure it out. You can say, because you don't make it tomorrow, it's your choice. But ask that question today. Answer immediately. But the journey will be interesting. The journey will accomplish a lot of it. It doesn't matter. But answer, you must ask. This is my advice to all young people and faculty. Find out what you want to say against your biodata. Excellent, very nice. Thank you for talking. But actually, if you ask me the social problem, where I was the chairman of the Antibiotic Resistance Committee of Pharma, I gave a report for the collection. Now, this year, I wrote
the report on which Professor Ganguly was the chairman.
We are now working. I was the chairman of the report. Okay, now it will be implementation of the report. The major problem is America has no infectious disease, but has largest epidemic. And this is because of the farm animals. India actually follows that. So farm animal is the base. So how do you prevent 2 generations of antibiotics gap between the application in farm animal and the human? Not to allow all the especially all the molecules like this is our data. The rest goes to filters. And here we are doing that. So human consumption is not even 1% of the antibiotic production. So that actually goes. So it's a social issue. It's not a house. It's a ministry as well. Right? But understanding what is
So I will.
Excellent lecture. Any questions from the audience?
Have you done any comparative analysis of antibiotics in the available market?
No. Basically, in the lab, a strip is there called E strip that is being used. And in market, I don't know that what is their level of concentration. Okay, because when you buy the strip from there is a company called Quince, and it's hugely costly. A pack of 10 strip costs almost 30,000. And when you go to a pharma shop, you can get antibiotics maximum in 10 rupees. So that is the level of impurity.
Yes, you said that you could find some amino acids which are relatively binding site and they may be responsible for this resistance.
No, it was opposite. We could.
Could you find particular patterns in that? What all amino acids were there?
No.
Was there any specific group of amino acids which were relatively binding?
And there are few mutations, but that according to our docking studies, that mutation was not contributing to the hydrolytic factor.
Any other questions?
Is there any database available that can be used by common people so that they can get benefit which antibiotics should be taken at a like what extent it goes? In day-to-day life, we are taking so much of antibiotics, so anything
I don't think an antibiotic is a over-the-counter drug. When it is proper physics, then only prescribed.
Any other question from the audience? Any no question? And we have tea after this. 11:40, we should be back by 11:40 sharp. 10:11, yeah. So as to be on time. Yeah. Sorry? So we'll depart for the tea now and we'll be back at 11:40 to be on the time. And the tea is being served in the glass store room next door. Rahul? Hi. Oh, good to see you back again. Right. I'll catch up with you. You're around for the whole day. Excellent.
How are you growing?
The first one.
के चल रहा है
हाँ चल रहा है. इसको प्लीज बंद करो. And next session will start with accounting.
Hello. Good morning, everybody. So our next speaker is our very own Vibhor Kumar. So before that, who am I? So I'm Debarka Sengupta, an assistant professor of the Department of Computer Science as well as Center for Computational Biology. So it's my pleasure to introduce my colleague Vibhor Kumar, who is our next speaker. Before IIIT, so he has recently joined IIIT. Before that, he was a research scientist in the prestigious Genomics Institute of Singapore, where he in fact started as a postdoc, and he worked there for nearly 7 to 8 years, I guess. And then he also worked in a company before that in the area of personal genomics in Malaysia. And he did his PhD from Helsinki University in 2007, where he solved the LDL structure in nice state at human body temperature. And he did his master's from IIT Delhi. So his research interest is in the domain of chromatin states and gene regulation. So he's also interested in clinical sides of all these things, including cancers and other stuff. So very warm welcome to Vibhor Kumar. I think he can introduce himself briefly too, if I missed something. Thank you.
Thanks, Devanta. So I hope you can hear me. Okay, can you hear me now? Okay, so welcome to my talk, and so I will not be able to visionary like Professor Chai, but I'll give some idea that you could think about some visionary projects. So nowadays there is a lot of hype about next-gen sequencing, and next-gen sequencing has generated a lot of data in different kinds of fields like epigenome, transcriptome, exome sequencing, and de novo sequencing of the whole genome. Now I'll go to just basics because I know something about computer science. So we see that we have heard about DNA. So actually DNA is actually wrapped around nucleosome. And nucleosome is because it consists of 4 histone protein molecules, and it is wrapped, and then there is a extra wrapping, and this way the DNA, the 2 micromolar DNA gets wrapped in a micrometer of nucleus. So you can see it has a bead and thread structure. This is called chromatin. And the proteins that are bound around chromatins and the DNA itself, and even RNAs, they all are called as epigenome. So people are trying to study epigenomes. What's the benefit of epigenome study? So the thing is that we have transcriptome, that is gene expression. Okay. Then we have epigenome. So epigenome is the gateway for transcriptome because epigenome allows, actually, it is it's a mediator of different kinds of phenomena like DNA replication and gene transcription and different kinds of like DNA breakage interface. So the promise of epigenome is great. For example, let's see an example here. So these both girls are twins. They have the same DNA, but one is fat and one is small. And recently, there was a paper in Cell in which authors have shown that one epigenetic modifier, TRIM28, if it gets upregulated and downregulated, it actually decides, predisposes a person for obesity. And similarly, for plants, epigenome has a great promise. So one enzyme for DNA methylation, if it is knocked down, the size of the rice plant decreases. So epigenome is a gateway for multiple things, many kinds of phenomena, so we cannot ignore it. So, and another promise, and this is a promising result from epigenome study. So what you see that autistic disorder it's called autistic spectrum. So there are different values of autism. And there's so much heterogeneity that drug companies have almost refused to actually do research in the field of drug development for autism. So there are antibiotic, hypothetism, there are different sources of autism. So I and my previous boss, Shyam Prabhakar, in 2009 we had a vision, and with one collaborator in US, that we were trying to study histone modification. Then we thought, why not we do kind of do some cohort study and try to actually correlate differences in the histone modifications at different locations of the genome to the underlying sequences. So then other group members joined, and we finally published one paper in Cell in last year, 2016. So it took 7 years to actually writing the grant, getting money, and wrapping up everything. And I was there from the start to end. So what we did here, we got brain samples from US, and we took 3 regions from the brain: prefrontal cortex, temporal cortex, and cerebral of autistic patients, non-autistic patients. So these are biopsies. These are not biopsies. These are like they have already dead. It's post-mortem. So, and we generated epigenome data. We took one acetylation. It's called STK27 acetylation, which marks active regions in genome. And then so there are a lot of speculations that there is so much heterogeneity in autism that you will actually not find any differential peaks. So what we found, in prefrontal cortex we found like 5,000 differential peaks. In temporal cortex we found 7,000 differential peaks with significant p-value. And just to prove that this is not nonsense, this is not false positive, we had set up positives and negatives, like autistic patient sample, non-autistic patient sample. We randomly shuffled them 1,000 times. So this is called permutation test. We did permutation, and on average, the permuted samples had very few number of differential peaks. It means it is the phenotype of autism that is causing this differential. So this is not false positive. And this gives a solid encouragement for drug companies that there is something consistent out of the heterogeneity in which we can do research and make a drug. So if it was really heterogeneous, no chance of success, we would not have got that many differential peaks. Now, see, there is heterogeneity in autism. So we took 2 different kinds of autism. One in which there is duplication in one part of chromosome number 15, and there is idiopathic autism. So when we took the differential peaks, we saw that these both different kinds of autism share same differential peaks. So the mechanism, one is having a CNV, duplication. The mechanism is epigenome, and it is same mechanism. So this inspired me to actually, why not we can do the same thing in cancer? In cancer, we have a lot of mutations, CNVs and everything. So I wrote a grant on cancer stem cell epigenome, and I got funding of 80 lakhs. A young investigator grant in Singapore, but I came back. But anyway, I did a lot of data. I was analyzing here, and so the motivation is that we have 6-7 cell lines of cancer, and they may have different mutations in CNVs, but the mechanism could be same through the epigenome. So we are talking about personalized medicine and targeted therapy and all that. If we include epigenome, we could actually simplify this problem. So it could be personalized epigenome-based drugs. So I have been talking about epigenome, but how to actually profile the epigenome? So one core part of profiling epigenome is called ChIP-seq. So what we do in ChIP-seq is that suppose we have a DNA wrapped around nucleosome where the histones tails are hanging out, and the histone tails have some modifications, and we want to profile the modification in which part of the genome this modification is happening. So we get an antibody for that modification, we pull that, and we cyclate the DNA into small fragments with the histone modification, and we pull it, and then we put it into the machine, sequencing machine, we get small tags. And then we get tags from both sides, from both the strands, positive and negative strands, and we map it to the genome. And the count of the tags actually decide the intensity of that modification or any transcription factor that is binding there. So we get a linear signal. So no wonder I am like signal processing guy working on genomics, because this comes to signal processing. Yes. Yes, epigenome is you have DNA wrapped around nucleosome, and we have proteins binding. So all the protein complex and the DNA itself, this is called the epigenome. Okay. So we have genome sequence. That's just genome. M complex with different proteins. It's called epigenome.
DNA, genome sequence DNA is static information. The information that is dynamic part of it when undergoes humanity code is static. The regulatory and control is
So, see, there are different kinds of epigenomic assays. So basically, this is what I explained. What is epigenome? So there are 130 histone modifications. So there are 4 kinds of histones in nucleosome. And so far, what is known that there are 130 different kinds of histone modifications. Okay? Modifications means they have acylations or methylations in the N terminal or the C terminal of the histone. And they cause they have a lot of regulatory effects, because these modifications recruit other enzymes to do a modification in the DNA or the complex, DNA protein complex. Similarly, we have more than 2,000 proteins which are binding to the DNA. So this is the dimension of the epigenome. So you have 130 plus 2,000 plus there is also there are other kinds of assays which says that okay, the DNA is open here and packed here. So they get nucleosome packed here and they don't allow any transcription factors to bind. And this is open, it's called euchromatin. So, and here the transcription is allowed, transcription factor can bind. Okay? So this information can be tried using different assays called ATAC-seq, DNA-seq. It's called open chromatin assay. So you can see the dimension. So if you take the DNA as like 33 billion, then you can have, or if you can bring the whole genome into like few million beans, then at every bin you have so many dimensions. So what is the histone, what is the level of this histone modification, what is the level of this transition factor, and what is it open and not. So you see the dimensionality of the epigenome data. So that's why it sometimes starts going into big data. So common trends that is following in the epigenome studies is that suppose we have a gene, so we have promoter, and the gene is being transcribed in this direction. So we have enhancers. So enhancers are the regulatory elements where transition factors bind or the proteins bind, and then they excite the promoters to start the transcription. And we have insulators which actually block the effect of repression from one region to migrating towards other region. And so far, there are different histone codes for active promoters and active enhancers. So basically, for active promoters, we say that okay, STK27 installation and STK4ME3 is present. Active enhancers, STK27 installation is present. Poised region in the epigenome means they are poised, and when excited, they will get activated faster than those which are silent. So these are poised regions. And then we have repressed regions. So basically, the repression is that something has to be done to remove the repressive complexes so that gene transcription can go. And then we have gene elongation mark. So when the gene is being transcribed by the altered complex, there are different histone modifications which accompany the transcription. Some of them actually follow the transcription, and some of them encourage the transcription. And there are different consortia across the world who are investing like millions of dollars in this. And so we have ENCODE, and then we are part of IHEC, International Human Epigenome Consortium, with which we published the autism study. And then a new kind of study is coming, that is epigenome of plants. Still a completely new field because of the promise of epigenome. So far, actually what people have been doing is they have been studying very few histone modifications because they're scared of the dimension of the data. It is not possible to actually profile every histone modification. So in 2009, we started one work that why not we find the most relevant histone modification that is useful for active enhancers. So we profiled, so we randomly chose the regions from the genome, and we tested doing an enhancer assay on CD4 plus T cells, and we got a list of positive enhancers. And then luckily we had 40 to 45 ChIP-seq data, and we did machine learning, and we found that the ChIP-seq data of H2BK20 installation was identifying the active enhancers more efficiently than all the others are. So this histone modification has not been studied properly, and that's why it was very exciting to us. So then what we did, we took B cells, we profiled H2BK20 installation. So basically we see every line here is a promoter. And this is the signal of different histone modifications. We clustered them. When you see that only promoter 1 class, P1 class, promoters have H2BK20 installation. And all the others have STK27 installation. And when we take the gene expression, when you see that the promoters which have STK27 installation, all of these have high expression. Okay. But when we take the cell type specific expression, that for every gene, we divide the expression by the expression across different cell types, then we get the cell type specific expression for every gene in B cell. When we see the cell type specific expression, we see that the promoters which have H2BK20 installation, they have highest cell type specific expression. So this is a new mechanism which was not discovered so far. And not only this, we did independent tests. We took promoters and we sorted them according to different histone modifications or chipset levels, or different transition factors level. And what we found that the top 1,000 promoters, we just took top 1,000 promoters for different marks. And we saw that promoters with H2BK20 acetylation had the highest cell type specific expression. So, coming to this, what is the use of this cell type specific marker, by the way? See, cell type specificity has a big role to play in cell identity, species delineation, and of course housekeeping genes maintain the simple housekeeping function in the cell, but the cell identity is maintained by the mechanism of cell type specificity. And that's why this is a big role to play. We have more than 200 cell types. How it is happening? So there are still some hidden mechanisms that we are trying to reveal. So what we did is that to see one benefit of cell type specific marker, we took GWAS SNPs. So SNPs which have been associated previously to diseases. So for B cells, we took autoimmune disease and their SNPs, and we said, okay, the mutations associated with autoimmune disorders, how much of them are being recalled by peaks for different marks? And what we found that H2BK20 acetylation has the highest overlap for the autoimmune disorders in cell. This is the benefit of cell type specificity. Not only that, then we exploited this result. And we took a rheumatoid arthritis. This is also one kind of autoimmune disorder. So there was a paper published on rheumatoid arthritis with big data. So they had two kinds of data. Smaller data from Europe had 2,800 to 780 rheumatoid arthritis patients and 4,000 control. Then they had meta-analysis where they had multiple populations, ethnicity, and there they had 29,000 rheumatoid arthritis cases and 73,000 controls, and they profiled the mutations in those. So what we did, we took the association studies from the smaller set. So the SNPs or the mutations that were associated with rheumatoid arthritis from the smaller set, we took them, and then we plotted them according to p-values, suppose. And then we took this as a kind of test. So we took the training set from here, and when we plotted according to the p-values from here, for the p-values here, we didn't see that correlation. But then I ignored the p-values completely from the smaller studies, and I sorted those mutations based on the level of H2BK20 acetylation. That's it. What we see, then when we sorted them, the meta-analysis results of p-value were highly correlated with the level of H2BK20 acetylation. So basically, cell type specific marks can help to identify the most relevant mutations for that cell type. Like autoimmune disorder is associated with B cells. And if we have profiled the cell type specific mark, then we can add confidence to the association studies. Now, this is also a very beautiful result. What we did is that, okay, so we took 16 histone acetylation data across an IM190 cell. We saw the same result. Whenever we had H2BK20 acetylation, those promoters had highest cell type specific expression. And then we took the chromatin-chromatin interaction data. So basically, the chromatin, the whole, you can say, epigenome, or it's kind of chromatin, it folds, scaffolds, and interacts with each other in the 3D. So it is like this. Okay? So So this could be like DNA, bead and thread structure. So using the 3D contact information, what we found that regions, promoters which have H2 BK20 acetylation, they have the highest number of contact with enhancers. So basically the enhancers regulate the cell type specific promoters and they have H2 BK20 acetylation. Not only that, when we look at the promoter-promoter interactions, so now I'm talking about 3D regulatory factories. So chromatin folds, so basically in the nucleus we have different regions. We call them a 3D regulatory factories where chromatin interacts to produce different copies of RNA. What we see that these promoters, IP1 promoters, they are interacting with each other more than the other class of promoters. Similarly, the housekeeping promoters, IP3 and IP4, they are interacting with each other more than the other class of promoters. So you can see that there is a regulatory factory. So one kind of factory will produce one kind of genes, like genes associated with autoimmune disorder, and housekeeping genes somehow interact with each other more. So this is the insight that we get from normal plants. Okay, so now I will come to what are the emerging trends in epigenomic study. So you might have heard about genome-wide association studies, EQTLs. So what we do in EQTL is that we correlate the mutations, the alleles at one mutation to the gene expression, to the nearby gene expression within 100 kb. And we say that if we find a correlation between a SNP allele to the gene expression, we call it as an EQTL. And for that, you need to either profile the mutations separately, or you need to sequence the whole genome individually. And there are different databases where you can get this information. But then we came out, with other solution that if we are profiling the epigenome, profiling deeply, limit like more than 50 million tags, then we can, using the same tags, we can get the mutations at the peak. So we can get the allele information of the mutations at the peak using the same tags, which is used to call the peaks or call the signal that this protein is binding or not. So for autistic disorder, we did the same thing. And this we call it as histone acetylation QTL, HAQTL. So for this, we don't need to sequence the genome. We just need to do the chip-seq profiling. We don't need to de novo sequence the genome or do a mutation profiling. We can derive the information about a certain mutation from the chip-seq tags themselves, and then correlate that allele information with the signal intensity of the histone modifications at that point. For example, what we found is that at this location, at this SNP, we found that GG allele has more level of HTK27 acetylation than the allele with CC. This is called histone acetylation QTL. Now, what is the benefit of correlating histone modifications to the underlying genome? Because people can say that finally I just want to study the gene expression. I don't care about modification. So the reason is that what I showed in my Nature Biotechnology paper is that when you take promoters and you take the underlying sequences of the promoter, they are more correlated with the histone modification than with the gene expression. The problem with gene expression is that there is post-transcriptional regulation, and then there are other mechanisms which actually blur the information. So, and that's why when we try to model the histone-based transcription rate, so I made a tool to predict the transcription rate using histone modification and promoters. So that I call it HPT. HPT is more correlated with the sequence motifs and promoters than the gene expression itself. And that's why for QTL analysis, you get more number of QTLs using histone acetylation than using gene expression because of the noise, the measurement biases in the gene expression. So now we are in a mission to actually find GWAS SNPs and find the actual causal mutations using HAQTLs. So it's not possible for all GWAS SNPs, but as we already know that 80% of the SNPs that has been associated with diseases are actually non-coding. So they could be enhancers, regulatory regions, and that you need to profile everything. So why not correlate the epigenome directly to the underlying sequence and actually find the true mutations? Maybe the association studies would have brought some mutations, okay, that this mutation is associated with certain disorder, but actually that's not true because there is another mutation line which is a linkage discovery or having the same correlation or coevolution, which is actually the true causal state. So this is the promise of HAQTL. Now the next emerging trend that is coming is single cell epigenome. So we are not there yet, but let me explain to you the profit of single cell epigenome. So it sounds crazy, like producing more data, but we have experienced it. So what we did, I and my colleague Devarka, we were analyzing data from Professor Ramanuj Dasgupta at GIS Singapore, and these are, so basically every row and every column here is a single cell, so every dot is a single cell. So we clustered the single cell based on this gene expression only, okay? So it looks like, okay, this is one cell type, like all these cells are of one type. So basically this was done from 15 cancer cell lines. So we have in one cell line you can have multiple cell types. Okay? So we merged the data from 15 cancer cell lines, and so it looks like this is one cell type. But when the drug is applied, they all show heterogeneous response. So what is happening? By gene expression they look like same, but when the drug response is different. There we need single cell epigenome. Because epigenome is poised, epigenome has information about the history of the cell, or the origin of the cell, and that decides the trajectory of the cell. Okay. So cells may look like having just epithelial characteristic, but one of the cells are actually is poised towards EMT, and one of the cells will remain epithelial. But gene expression of both will look same. Both will look epithelial. And that we have already shown, and submitted to Nature Genetics. So it's already in review. So, but we have not profiled a single cell epigenome. What we showed is with bulk epigenome. But anyway, so there are publications which claim that they can do single cell epigenome profiling, the single cell ATAC6, single cell DNA methylation. So, but the problem is these data are so sparse. Because for a cell, in a cell, you can have multiple copies of RNA, but for DNA is just one copy. Right? So, profiling the epigenome, that one copy of DNA is a bit tough, but there are technologies coming up. So, I acknowledge for my presentation work by my previous boss, Dr. Syam Prabhakar, the director of GIS, and my collaborator Masafumi Muratoni. He is a professor now in Tokugawa University, Japan. And my other collaborators, Nimla, Sanbenji, Jeremy, so they work together now. They have all moved to different countries like India, Japan, and of course Shipra, who has been working hard to organize this symposium. And I was there, another faculty member to like Thank you. Any questions?
Can I get the mic?
Are there human models available for autism which Epigenomics have been rated and gone to?
Yes, there are some animal models, but they are for particular kinds of autism. Like this. So it is possible, but only for certain particular kinds of autism.
How specific can targets in Epigenomics be? Are they going to be what is the expected?
So, from our human-based study, it looks like there is some common mechanism that is causing this syndrome. Because phenotypically, there is some overlap between autistic patients, but the source might be different.
Right now, not that much successful because
so if we take out a cell from the body and do some modification in the DNA to treat some disease and then we put like this cell. So the cell does not feel more present, the cell is forgotten this history. And you just said that epigenome somehow remember the history. So can epigenetics help in that?
That's a very good question. And actually, people are trying to do that. So when you take, so for epigenomic studies, what we have been doing, what we have been thinking, and I have planned some studies here in India also. So directly from the patient. See, culturing the cells after taking out may actually learn the information. But it may have some memory of previous, but current conditions. So there is a memory that is in the police region, and there is an active transcription. Though they both might be different. So it's a good question to be explored. That if you culture the cells out from the real samples, that is remembers and can be retrieve any information that why this person got this. That's very good. That's a very good, like, innovative question actually. Yeah.
I think it's very nice. You have actually covered wonderfully large matter. As far as animal model is concerned, the greatest animal model of epigenetics is mule and hen. Mule where mother is a father is a boy. The other way, and the genetic composition is the same. The process. I think what would be interesting, I tell you where the sickle cell data has some anxiety at stake. Because you know very well, your behavior at home is different than your behavior in the office. And your behavior in presence of your boss is different than your subordinate. So a single cell behaves depending on the cells around the car in a given case. So therefore, if you isolatedly see without a context, then you lose the system. So my request would be, when you look at a system, then you have to see many cells taken out of liver and study them collectively. So you have 100 cells taken out of liver,
Right.
and then see collectively how they are stochastically distributed in various ways.
That's true.
And it is this communication together makes the tissue. The tissue is again multiple cell types comes together. Finally an organ. So liver knows what to do, a kidney knows how to filter. But each of the cells in harmony doing the job. There is fantastic advantage of doing what you call breakdown analysis, right? But I'm really happy to see you doing a very interesting, you are a fantastic.
That is true. So epigenomic profiling from single cells from the biopsy sample. Nobody has done it.
Yeah.
They're still thinking about it. Because it's very hard.
I know it very well. It's very complex.
Single cell epigenome from biopsy sample is
The biggest example that you are doing is cell differentiation. You know very well that a stem cell gets differentiated to different cells. That's where epigenetics comes into play. Because OCT4, SNCF2 and all these are depending on the methylation status of those two histones. You can switch this way or switch this way. So I suggest if I'm going to invest my money on you, and I will like to invest time where you look at stem cell differentiation associated with single cell approach.
In the microenvironment.
That's where the microenvironment, with each cell is committed there to become a differentiated cell. Whereas when you are already taking a tissue, cancer tissue and all, they get hodgepodge. Number two, don't work on cell lines. I tell you why. Cell lines are polyploid. Minimum is 70 to 90 when you work on cell lines, immortal cells. They're polyploid. They have nothing to do with you. They're far away from you. You will get data. It will get published in Cell. 20 years, 10 years later, nobody will cite you. You are gone.
So, but one thing,
So my question,
you are right.
You must have Bob Langer is to tell, you must have an unfair advantage of doing science.
That's true. You are right. But what you're saying, I can object to it because, we had patients like cells taken from biopsy from 2-3 patients. Right? One cell line never went to EMT. Cells from one patient never went to EMT. Cells from other patient went to EMT.
Correct.
And in the cell line. So, cell line culture of cancer can actually give you some insight over the mechanism.
Absolutely, with respect to MTFR, targets,
Yes.
precision medicine, there's application.
Yes.
I'd say since single cell is the fundamental stages, the early stages of development.
Yes.
The biggest role a single cell plays is different.
That's true.
Right? Subsequently, is there, not there, methylation, expression level. The problem is in the expression, only when you look at chip data or things like that. We don't know how miRNA comes in and blocks the protein production. So finally, protein production is the what finally will do the function. No? So the difficulty of this, at this moment, protein on single cell is impossible. Correct? We have not reached that level of mass spectroscopy.
There is some research going on.
It is reaching, but it has not reached. Because your gene may be expressed, but miRNA may come and block it. You don't get the protein. If there is no protein, no OCT4, no function, it will stay on as a one cell. The moment you got the protein, it will divert the whole chain of expression. So finally, few transcription factors, which is fantastically you can measure this thing, and you have done a wonderful job of analysis. So I'm saying, take something where actually it is doable today. It is, but that's what the biggest challenge. Choosing that, I think you are already there. You have fantastic tools you have. So my request to you would be that don't get, cells are so heterogeneous. You will get data and then you spend all your time spending on it. So paper bhi ho jayega. But problem solving. So you try to solve, because you are a fantastic trained person. You have understood the whole system very well. So the biggest challenge today, how do I revert back a differentiated cell back to cell? Or how do I have a stem cell taken from blood and induce it to become a patch for Alzheimer patient? So your cell, I take stem cell from bone marrow, I do a epigenetic modification and create a patch to the brain.
This is exactly what I'm thinking on there. Right? But there is one paper already in Nature Methods where they have published like set of transcription factors like by computational analysis, which can be used to transform any cell type to any cell type. It's already published, but it is using Yamanaka-like factors, like through viral vectors. I am day and night thinking that can we have an external signaling molecule that can differentiate one cell type to any single cell type. I am thinking day and night since 1.5 years, but I don't know when I will do this.
I'm very
But I am looking for a talented guy.
I'm very selfishly interested because I know soon I may get Alzheimer.
Yeah. Well, I'm looking with the talented guy for
Waste
this.
of time. We are just including this lecture. Thanks. So.
Hello. So thank you. Next speaker is Dr. Dinesh Gupta, who is a group leader of Translational Bioinformatics group at ICGEB and the coordinator DBT Bioinformatics Infrastructure Facility at ICGEB. Dr. Gupta received his MSc and PhD degrees from All India Institute of Medical Sciences, New Delhi. He is a member of DBT Task Force for Bioinformatics. Till recently, he was the chairman of the DBT Committee for Promotion of Biotechnology, Related Research and Training Activities. He is an academic editor of Class 1 journal. He received a WHO capacity building grant to develop regional research and training center at ICGEB for conducting bioinformatics research and providing bioinformatics training to Southeast Asian researchers working on tropical diseases. He has been on a capacity building fellowship to the University of Pennsylvania and Wellcome Trust Sanger Center as a visiting research fellow, where he made extensive contributions towards the development of Plasmodium Genome Consortium resource site www.plasmodb.org. He is a member of different professional societies like ISCB, APBIONet, and IPS. The scientific interests of his group include the development of computational biology tools for translational mathematics. The group is also interested in using artificial intelligence-based methods to solve high throughput data classification problems. With that, I'd welcome Dr. Dinesh Gupta on stage and to do his lecture. Thanks.
Good afternoon, everyone. Thank you, Javakar, for a nice and elaborate introduction. Though I wasn't expecting that you will read out the whole thing. Thanks anyway. I thank Dr. Raghav for inviting me for this symposium. I'm really enjoying it since morning, especially Dr. Sanchari's lecture and Manish's, and then followed by Dr. Kumar's. So since the time is less, I will try to shorten my talk, though I don't promise, but let us see how it goes. So today I will be speaking on high throughput data analysis for cancer diagnostics. I will be talking about a little bit about
percent is what it is. For example, if you just take this class and ask, how many of them are useful? So you get 100%. Okay? But if you take the work probability by now and pick up and election campaign somewhere in Vietnam. So when the features, when the sample set becomes larger and larger, many features will start appearing in the machine learning. The question is, if you are a machine learning and successful, how do you get homogeneous data from which you learn and then go through heterogeneous data and say connect it? So therefore, it is obvious that if you use a large data set, you will never be able to reduce your dimension.
Absolutely right.
Supposed to be